being slightly late for the defense convocation and travel around Moscow is sort of unpredictable these days, unfortunately. Presenting the jury members, and I'll start with our international colleagues. So Dr. Edward Polt is Associate Professor of the Faculty of Medicine and Health Science of the University of Nottingham in the United States from the University of London, and technology, in particular, he works on the involvement of CRISPR systems in bacteria and the mechanisms of DNA repair. Professor Francisco Juan Martinez Mojica is a full professor at the University of Alicante in Spain, where he received his PhD some time ago. And he is well known in the CRISPR area because he was one of those people who have actually discovered these systems. And currently he works on a fundamental molecular basis of the CRISPR-Cas, specifically in CRISPR pathogenicity association in Escherichia coli. Dr. Peter Finneran is a professor at the Department of Microbiology and Immunology at the University of Ottawa. He works on regulation of CRISPR-Cas systems, and so works a lot in like public area, in educating students and industry about CRISPR-Cas systems and their applications. Now, the Caltech members of the jury are Dr. Ekaterina Khraneva, an assistant professor at Caltech. Her main interests are in various PAMEX technologies. She worked on alternative splicing for a while, then on the PAMEX, and her main interests these days are on three-dimensional structure of chromatin and its relation to aging and various processes happening in humans. Professor Yuri Katalevtsev, a full professor at Center for Neurobiology in Caltech. His main interests are in molecular genetics of disease and gene targeting in embryonic stem cells. And myself, I'm a full professor at Caltech as well, and my scientific interests are various aspects of molecular evolution, in particular evolution of bacterial genomes, systems of transcriptional regulation in bacteria, and so on, and also in evolution of liquid carriers as well. Our candidate today is Elena Kurilovich. She started working with Professor Severinov about 10 years ago. She graduated from Moscow State University. Molecular aspects of CRISPR adaptation, the direction of CRISPR-Cas systems and genome repair, and during her study at the previous program of Caltech, she also spent considerable time in our academic mobility program in Professor Timron's group in Tel Aviv University and Professor Vestra's group. Our scientific advisor is Professor Konstantin Severinov, the head of the Russian biotechnology campus, which aims at sequencing 100,000 genomes of representatives of the Russian and all of, but historically he worked on microbiology in analysis of CRISPR-Cas systems, their interactions with phages, and various research modification systems and other modules with which bacteria fight the phages. That I apologize once again. I'll be on site in a couple of minutes, hopefully. And the floor is Elena's. Benita.
So, Elena, the floor is yours. Please unmute the mic and start the presentation.
Okay, yes, do you hear me well? Good. Good morning, dear jury members and colleagues. Today, I'm going to talk about my PhD thesis project, the role of genome maintenance proteins in primed CRISPR adaptation by the type I-E CRISPR-Cas system. The work was done under the supervision of Professor Konstantin Severinov. Sorry, maybe I pressed the wrong button. Let's start from the introduction. First, I'll tell briefly about the type I-E CRISPR-Cas system of E. coli. It consists of a CRISPR array representing, oh my God, why? Sorry, Elena. It's some problem with the presentation. I don't know why. So it consists of CRISPR array representing identical repeats with unique spacers between them, and CRISPR-associated Cas genes, namely Cas3 encoding nuclease translocase, 5 Cas genes encoding a multi-subunit cascade complex, and Cas1, Cas2 encoding CRISPR adaptation complex. CRISPR-Cas system is functioning in several steps. First, CRISPR array is transcribed into the precursor CRISPR RNA, which is then processed into several short CRISPR RNAs. When the foreign DNA gets into the cell which contains a sequence complementary to one of the CRISPR RNAs, it is recognized by the cascade CRISPR RNA complex, and then Cas3 is recruited, which starts the DNA degradation. This process is called CRISPR interference, and also to be properly recognized, the sequence in foreign DNA, which is called protospacer, should also contain a proper protospacer adjacent motif of PAM. The products of DNA degradation suggested to serve as substrates for Cas1, Cas2 adaptation complex, which performs the incorporation of new spacers into the CRISPR array. Such interference-coupled CRISPR adaptation is called primed CRISPR adaptation or priming. The important feature of primed CRISPR adaptation that almost all new spacers correspond to the canonical PAM, which is AAG for E. coli, and originate from the protospacer region. And if the foreign DNA is not recognized by the CRISPR-Cas system, its fragments may still be incorporated into the CRISPR array by Cas1, Cas2 in the process called naive CRISPR adaptation. In this case, no other Cas proteins are required, but this process relies on DNA repair and replication. And in this case, only half of new spacers correspond to the canonical PAM. In our work, we were focused on primed CRISPR adaptation. And if we look closer, it is a multi-stage complex process. First, the priming protospacer, or PPS, is recognized. And primed acquisition complex is assembled, PAC, which consists of cascade, Cas1, Cas2, and Cas3. DNA strand which is recognized is called target strand, and another strand which is cleaved by Cas3 is called non-target. So then the whole complex translocates the DNA due to the Cas3 activity. The strands get separated, and Cas1 recognizes PAM complementary sequence TTC. And then Cas1, Cas2 complex bind the adjacent single strand protospacer. Cas1, Cas2 then anneals it to another complementary AAG associated strand, and then the prespacer precursors formed as a result of DNA degradation and bound by Cas1, Cas2 are trimmed to form prespacer precursors. And prespacers, and the prespacers are then incorporated into the CRISPR array in an orientation-dependent manner, so that the first nucleotide of spacer is the last nucleotide of PAM, and the resultant spacer is 33 base pair long. But still, it is not clear how exactly prespacer precursors and prespacers are formed. It is known that some CRISPR-Cas systems of type 1 encode an additional Cas4 protein, which trims 3' ends of prespacer precursors, thus processing the 3' ends, and Cas2 protein of Streptococcus thermophilus was shown to be fused to DNA Q domain, possessing 3' exonuclease activity. At the same time, E. coli lacks Cas4 or DNA Q domain in Cas2, and little is known about the processing of 5' ends of prespacers. So we suggest that additional proteins must be involved in primed CRISPR adaptation in E. coli. There is more evidence suggesting that genome maintenance components may take part in CRISPR adaptation. It is known that naive CRISPR adaptation relies on REC-BCD helicase activity, and REC-G and pre-A helicases has a role in primed CRISPR adaptation during lambda phage infection. And moreover, the components of the double-strand break repair system, namely REC-BCD, SBCD, and SBCB, or EXO1, frequently co-occur in E. coli genome with CRISPR-Cas systems. There are two main DNA repair pathways in E. coli. The first REC-BCD pathway is responsible for the repair of double-strand breaks, which can normally occur during the DNA replication at the sites of collapsed replication forks. In this case, REC-BCD binds blunt or nearly blunt ends, and upon facing a specific Chi sequence, it generates a free 3' end and mediates the loading of REC-A, which initiates the homologous recombination, thus restoring the break. REC-BCD complex possesses both helicase and exonuclease activities, and in REC-D mutant cells, REC-BC complex is assembled, which retains helicase but loses nuclease activity. If for some reason REC-BCD pathway is broken, it can be substituted by the REC-F or R pathway, which is normally responsible for the single-strand break repair. In this case, REC-Q is a helicase, and REC-J and EXO7 are 5' exonucleases, which may generate a free 3' end essential for REC-A loading. And in this case, F or R complex are mediating the loading of REC-A. Also, there are proteins SBCB and SBCCD, which are suggested to take part in DNA repair by blunting the DNA ends for REC-BCD loading. And SBCCD also possesses, also can cleave hairpin structures. The goal of the whole work was to study the role of different cellular genome maintenance proteins in primed CRISPR adaptation in the type I-E CRISPR-Cas system. And the objectives were first to check the requirements of different recombination repair pathways in CRISPR adaptation, and to study the impact of genome maintenance proteins on adaptation efficiency, to reveal the effects of these proteins on spacer choice and the generation of spacer precursors. The first part of the results is based on the publication in Genes 2019. In this work, I performed the construction of mutant strains, preparation of samples, and data analysis. The model system, as a model system, we used a self-targeting E. coli strain, which has Cas genes under inducible promoters and a spacer in a CRISPR array, which encodes a CRISPR RNA targeting a protospacer inside the chromosome in a non-essential gene. So when CRISPR-Cas system is induced in this strain, it leads to the inhibition of cell growth, and the analysis of total DNA sequencing in induced cells revealed an extensive DNA degradation in the region of protospacer. And the analysis of amplicons derived from the extended arrays revealed the hallmarks of primed CRISPR adaptation. Here, the graph shows the mapping of new spacers on the PPS region. The blue curve corresponds to the non-target strand, and red curve corresponds to the target strand. It appeared that almost all new spacers correspond to the canonical PAM and originate from the PPS region. And we see that spacer acquisition is bidirectional and orientation dependent, so that acquired spacers preferentially map to the non-target strand upstream of the PPS and downstream of it from the target strand. So we obtained knockout mutants of this strain by P1 transduction, lacking genes encoding different genome maintenance proteins, induced CRISPR-Cas system in these strains, and then did a PCR on a CRISPR array to detect CRISPR adaptation, and then sequenced new spacers and analyzed them. Here on the left, you see the result of the PCR on a CRISPR array in wild type and mutant strains. The lower band corresponds to the initial unextended array, while upper bands correspond to the arrays which acquired one or two more spacers. And on the right, we see the adaptation efficiencies calculated based on the intensities of the gel bands. We see that no detectable CRISPR adaptation is observed for REC-B SBCD or REC-B REC-J mutants. And we see that the levels of CRISPR adaptation in REC-J mutant is dramatically decreased, and the adaptation level for SBCD mutant is increased. Then we cut the bands corresponding to the elongated CRISPR arrays, extracted spacer sequences from the amplicons, and mapped new spacers on a reference genome. So here on each graph, you see the mapping of new spacers on the PPS region. Again, blue curve corresponds to the non-target strand, and red curve corresponds to the target strand. We see the gradient with the maximum peak at PPS gradually decreasing with the growing distance from it. On other graphs, blue and red corresponds to the wild type, while green and magenta show the results for mutant strains. We see that for all three REC-B CD mutants, the area from which spacers are acquired is extended from the PPS. And here are the results for other 4 mutants. And we also see that for SBCD mutant, this area is also extended, while there is no effect of REC-J and SBCB mutations. The pattern is also extended for double REGB SBCB mutant, but the result is similar to the effect of a single REGB mutation. Then we also assessed the precision and specificity of spacer incorporation in mutants. So there is always a small portion of spacers which are flipped or shifted several nucleotides up or downstream. And previously it was also shown that PAM sequences inside spacers are normally avoided. Here on these histograms, we may see that the percentage of spacers which are incorporated inaccurately or those with internal PAM is increased for REGJ mutant. So to summarize the results of this part, we demonstrated that REGBCD and SBCD affect the pattern of acquired spacers. So we suggest that these enzymes could affect the processivity of primed acquisition complex. We showed that primed adaptation efficiency is dramatically inhibited in double REGB SBCD mutant. And we propose the independent involvement and redundancy of SBCD and REGBCD in primed adaptation. We showed that the deletion of REGJ decreases adaptation efficiency and the accuracy of CRISPR adaptation, and showed that a double REGB-REGJ mutation abolishes primed adaptation, but REGJ deletion does not affect the pattern of acquired spacers. So we suggest that the pathways that involve REGBCD and REGJ are non-redundant and act on difference adaptation, and REGJ acts downstream after REGBCD. But still, the mechanisms behind this revealed interplay remained unclear. So we decided to study the roles of these proteins in prespacer generation. And the next part of the results is based on the publication in Science Advances 2022. Previously, Anna Shuraeva showed that prespacers formed during primed CRISPR adaptation in E. coli, 33 base pair duplexes with a blunt PAM distal end and with a 3' overhang on the PAM derived end with 3 or 4 additional nucleotides. And she used the FRAC-seq method. The procedure, during this procedure, short fragments are specifically purified from the total DNA, isolated from the cultures with induced CRISPR-Cas system. So we decided to check the roles of REGJ and REGBCD proteins in prespacer generation using this method. Here on the left, you see the coverage of the reference genome by the short fragments, and a zoomed in PPS region on the right. The prespacer-like fragments associated with canonical PAM are shown in purple, while all other fragments are shown in black. Here we see that no prespacer-like fragments are detected in double REGB-REGJ mutant, and the amount of these fragments is greatly decreased in REGJ mutant. We also can see that the prespacers are generated at greater distances from the PPS in all 3 REGBCD mutants. So these results are in a good agreement with the results of the previous work on these mutants. Next, to assess the prespacer processing, we determined all the chromosomal coordinates of AAG-associated protospacers located in the PPS area in the correct orientation, and calculated the distances from 3' and 5' ends of prespacer-like fragments to the boundaries of the corresponding protospacers. So here the histograms show the distributions of the distances 5' and 3' ends respectively in both strands. Here, positive values mean that the shift was beyond the protospacer boundaries, and negative values mean that the shift was in direction to the center of protospacer. Here, the arrows indicate the direction of the shift. We conclude that none of the mutations affected the processing of 3' ends, but for RECJ mutant, we see that most fragments, most 5' ends of fragments are 1 nucleotide longer than in wild type. While for RECBC, the effect is opposite, and most ends, 5' ends, are in general 1 nucleotide shorter. And interestingly, there is no effect of RECD mutation, and this is true for both strands. So we conclude that RECJ exonuclease activity and the RECBCD helicase activity are involved in the processing of prespacer 5' ends during primed adaptation. And then we hypothesized that in the absence of RECBCD and RECJ, other proteins, namely RECQ and EXA7, encoded by XSCA gene, respectively, could substitute them in their absence. So we constructed additional strains with double mutations, RECB, RECQ, and RECJ XSCA, and assessed the primed adaptation efficiency based on our high-throughput sequencing data. And indeed, we see that CRISPR adaptation efficiency is dramatically decreased in both double mutants compared to the corresponding single mutants. And also, using this method, we see that the CRISPR adaptation efficiency is also reduced in all 3 RECBCD mutants. And we think that this method is more sensitive than the one we used in the previous work based on the gel intensities. Next, the next question was if the reason for the decreased space acquisition efficiencies observed in RECBCD and RECJ mutants could be caused by the altered structures of prespacers. To test this hypothesis, we did oligo transformation assay, oligoelectroporation assay. So we transformed E. coli cells expressing Cas1 and Cas2 from a plasmid by the model prespacers of different structures. And we observed that both oligo-acquisition efficiency and the accuracy of incorporation were not affected by shortening or elongation of 5' ends of the model prespacers. So we conclude that this hypothesis was not right. But when we calculated the prespacer generation efficiency as a ratio of the amount of prespacer-like fragments in the PPS area to the amount of the fragments in a control region, it revealed that the lowered efficiency of prespacer generation is the likely reason for the reduced CRISPR adaptation efficiency in RECBCD and RECJ strains, and not the altered structures of 5' ends. Then also in vitro experiments were performed by Konstantin Kuznetselov using substrate which were 23 base pair duplexes with non-complementary 3' and 5' ends with a labeled 3' ends. And we observed that when RECJ was added with Cas1, Cas2, the substrate was trimmed to the prespacer boundary observed in vivo, and EXA7 produced slightly longer ends, and this is true for both strands. And we hypothesized that Cas1, Cas2 binds long prespacer precursors and with double-stranded regions, and then RECBCD can bind these double-stranded regions and wind them, and thus provide an SS for RECJ for trimming 5' ends. And to test this hypothesis, we used double-forked substrates with a 5' end on a palm proximal side annealed to the oligo so that the double-stranded region is formed. And we observed that when RECBCD or RECBC was added together with RECJ, to the Cas1, Cas2 bound substrate, each was trimmed to the proper prespacer boundary. So this experiment proved the hypothesis. To summarize this part of the results, we show that RegJ exonuclease and RegBCD helicase are involved in trimming of prespacer 5' ends during primed adaptation, and EXA7 can partially substitute RegJ. We show that also that RegJ, RegC, and RegB mutations decrease both the prespacer generation and adaptation efficiency, while double mutations, RegB, RegJ, and RegJ XSCA, abolish the prespacer formation and CRISPR adaptation. Then we also used a new method developed in our lab, which allows to detect half-integrated prespacers in vivo. Previously, it was shown that spacer integration in vitro proceeds in several steps. First, 3' hydroxyl attacks the phosphodiester bond between the leader and the first repeat, and the second attack happens between the first repeat and a spacer. And then double-stranded new spacer is formed, flanked by 2 single-stranded repeats. And here is a scheme of the procedure of our method for half-integrated prespacer detections or HIPS detection. First, the adapter with a free 5' end, the adapter with 5' overhang is added to the DNA isolated from the cells with induced CRISPR-Cas system. And this adapter anneals to all 3 5' ends in DNA, including the 5' ends of HIPS. And to amplify specifically HIPS 5' ends, to amplify specifically HIPS, we do a PCR with primers with a pair of primers, one of each annealing to the adapter and another one annealing to the CRISPR array. And depending on the location of the second primer, we can amplify top or the bottom strand integration products. This is then followed by the introduction of the second primer, indexing and sequencing. And here are the results for some of the mutant, the results for wild type and mutant strains. The graphs show the distribution of half-integrated products in the PPS area. We see that HIPS for the RegB, RegJ mutants were identified, though we couldn't detect prespacers for this strain. So we conclude that this method is more sensitive than FragSeq. We also see that HIPS distribution for the RegJ XSCA strain is very flattened and doesn't have a peak at PPS, which is well consistent with the previous results on this strain. And there is no effect in RegB, RegQ mutant, though we detected a greatly reduced CRISPR adaptation efficiency in this strain. And then we assessed the length distribution of HIPS. Here are the results for RegBCD and RegJ mutants. And these results are well correlated with the results of our prespacer length distribution. We see that here for RegJ, the HIPS are 1 nucleotide longer in general comparing to wild type, and for RegBC, they are 1 nucleotide shorter, while there is no effect of RegD. And here are the results for some other mutants. We see that the effects of RegB, RegJ are additive. So most HIPS are 1 nucleotide longer, like in RegJ, but at the same time, the amount of the shorter HIPS is also high. And also here, we see that the distribution for RegJ XSCA mutant resembles a non-specific background in agreement with previous results. And there is no effect of double RegB, RegQ mutation comparing to the RegB. And as a result of the whole work, we propose the following model of prespacer generation. So first, Cas1, Cas2 are bound to the selected protospacer determines the boundaries of a future prespacer by protecting them from degradation. And then the 5' ends of double-stranded prespacer precursors are trimmed by RegJ in association with RegBCD. And still a set of enzymes generating prespacer 3' ends in vivo is to be determined. And we also propose an additional role of RegBCD in primed adaptation. We suggest that RegBCD can catch up with primed acquisition complex, moving away from the PPS and displace it. And thus, RegBCD can limit the area from which protospacers can be selected. I would also like to briefly mention the work I did during my academic mobility in Exeter University. The results were partially published in Self-Hosting Microbe 2021. This work was focused on primed adaptation in Pseudomonas aeruginosa. It is a human opportunistic pathogen of a high clinical relevance. And it is particularly difficult to treat due to film formation and low antibiotic susceptibility and resistant development, of course. And combining phage therapy and antibiotic treatment was shown to be more efficient in clinical studies. At the same time, around 36% of clinical Pseudomonas aeruginosa isolates possess functional CRISPR-Cas systems. So we worked with a PA14 strain, which has 1F CRISPR-Cas system, and with one of the spacers corresponding to the phage DMS3 weir, with several mismatches supporting priming. It was shown previously that during phage infection, CRISPR resistance predominantly develops in minimal M9 media and surface modifications in rich LB media. The question was, what are the effects of antibiotics on the CRISPR-Cas immunity development during phage infection? The objectives were to conduct devolution experiments with PA14 infected by the phage in both rich and minimal media in the presence of different antibiotics, and to study the impact of antibiotics on the CRISPR-Cas immunity development. The experiment was done in the following way. Different antibiotics were added to the media at sub-inhibitory concentrations. Then the cultures were infected by the phage in 6 biological replicates. Then the cultures were daily transferred to the fresh media. The aliquots were plated daily to get colonies, and then the colonies were picked and tested for phage resistance. For the type of phage resistance by strict assays across wild-type phage and the one which encodes anti-CRISPR protein. And then the phage titers and cell counts were also determined. Here, the histogram shows the change in a proportion of clones which acquired CRISPR-Cas immunity, shown in blue, or surface modifications shown in orange, compared to the no antibiotic treatment. We see that all bacteriostatic antibiotics. Oh. Elena, sorry. Thank you. That all bacteriostatic antibiotics increased the proportion of CRISPR-Cas resistant clones after 3 days of experiment. And there is no such effect in presence of bactericidal antibiotics. To reveal the mechanisms of the observed effect, first we conducted a short-term assay for 3 hours to limit the possible effect of natural selection. And we observed that the proportion of phage-resistant clones that acquired new spacers in the presence of bacteriostatic antibiotics increased, and there is no effect for bactericidal ones. And also we conducted one-step phage growth assays to detect the production of mature phages. And it revealed that all bacteriostatic antibiotics greatly decreased the production of phages comparing to non-antibiotic treatment. So the results supported the idea that bacteriostatic antibiotics slow down phage development, thus providing more time for CRISPR-Cas system to acquire new spacers. And I also performed the experiment in minimal M9 media, and it revealed that bactericidal antibiotics have the opposite effect, and the proportion of clones which acquired surface modifications increased in presence of bactericidal antibiotics. But the mechanisms behind this effect are still to be studied. So here are all the conclusions of my work. First, primed adaptation efficiency in self-targeting E. coli strain is dramatically inhibited in double SBCD mutants, but not in single mutants, suggesting independent involvement and redundancy of REC-BCD and SBCD pathways in spacer acquisition. REC-BCD and SBCD affect the pattern of acquired spacers, which suggests that these enzymes could interfere with the processivity of primed acquisition complex. We show that the deletion of REC-J decreases adaptation efficiency and accuracy of spacers incorporation into the CRISPR array. We demonstrated that REC-J and REC-BCD are involved in trimming of prespacer 5' ends during primed adaptation. And we showed that EXA7 can partially substitute REC-J, but produces slightly longer 5' ends. And also, we showed that the exposure to bacteriostatic antibiotics promotes primed CRISPR adaptation by slowing down phage development in Pseudomonas aeruginosa infected by the DMS3 phage. And also show that bactericidal antibiotics inhibit CRISPR adaptation during phage infection by a yet unknown mechanism. The results of my work were published in 3 journals and presented on 2 conferences. And I'm grateful to my supervisor, Konstantin Severino, for his great support and guidance. And I am particularly grateful to Ekaterina Savitskaya, who is not longer with us, but she initiated the project, which resulted in the current thesis. And I'm grateful for her support throughout the whole way. And I'm grateful to Anna Shiraeva for her huge impact in this work. I would also like to thank Professor Edze Vestra and Tatiana Dimitriou for kindly hosting me in Exeter University. Thank you.
Okay, thank you. And we can now move to reviews by the jury, probably in the same order. So let's start with Professor Bolt, please.
Hello again. Thanks, Elena. Firstly, congratulate you on a fantastic body of work. Some great research there. I wanted to really just focus in on my expertise is homologous recombination, really, not CRISPR systems. So I wanted to focus in more on the DNA repair and homologous recombination aspects of the work. The first question I wanted to ask is, how do you envisage, so your data shows that REC-J and REC-BC probably are trimming ends of protospacers, etc., and other RECs may be involved. But how do you envisage that the protospacer is captured by Cas1 and Cas2 in the first place before this interrelationship occurs between all these different nucleases?
Yes, so it's not still clear what is the first. It's like DNA degradation occurs first, or Cas1, Cas2 binds the substrate first. But so it seems that Cas1, Cas2 first bind one strand of the future protospacer precursors, then anneals it to another strand, yes, then already yes, and then the formed double-stranded substrate is already trimmed by REC-BCD and REC-J.
Okay, so there's a lot of nucleases in this scenario. Where
Yeah.
does Cas3, I think you focus on primed adaptation,
Mhm.
as I tend to look more at naive. So forgive me if my question is naive, but where does Cas3's role as a nuclease fit in with all these other nucleases, REC-J, potentially REC-BC, although I doubt it? What's the role of Cas3 and the relationship between that in primed adaptation and the capture of the initial protospacer and then its processing to a pre-spacer?
Yeah, previously, so we have data which points that to the fact that REC-BCD can take part in DNA degradation during primed adaptation. Indeed, we don't have such such a result for REC-J, but for REC-BCD, yes, we indeed we observed that REC-BCD mutation affects the gap in the protospacer region during primed CRISPR adaptation. So we suggest that REC-BCD can take part in DNA degradation.
Okay, so I'm still trying to get my head around something more generally about how in primed adaptation, Cas3 is generating single-stranded DNA, and yet the pre or protospace is double-stranded DNA. Can you give me the latest clarity on how that comes about? How can you reconcile the various nuclease activities, which are all single-stranded nuclease activities, with the formation of a double-stranded pre-spacer or protospacer?
So anyway, it seems that first Cas1, Cas2 binds single-strand DNA, and only after it binds single-stranded DNA formed as a result of Cas3 activity, it anneals another strand. So
that's interesting, because I know some of the work that you've been involved in suggests that, and so that I wanted to bring in a type of DNA repair that you haven't really covered, and I'm quite intrigued about myself at the moment, particularly in bacteria. What do you know about gap repair, replication-dependent gap repair? You mentioned a bit in your talk about REC-FOR, but that doesn't appear in your thesis much, which was a bit disappointing for me. What do you know about gap repair, the functions of these enzymes REC-FOR, and how that relates to perhaps Cas1 capturing single-stranded DNA? Because I think that might get to the heart of the matter. So the question is, what do you know about gap repair, replication-dependent gap repair?
yes,
do you know, for example, I can help you out a bit here if you like. Do you know what genetic factors are required for in E. coli replication-dependent gap repair? Because they're very relevant to the work that you're doing.
Yes, I'm not sure that maybe this pathway can indeed take part in primed CRISPR adaptation in our case.
So then I would ask you, where does the single-stranded DNA come from? It comes from Cas3, does it, in primed adaptation each time?
Yes, like in the end, it was shown that Cas3 it degrades both strands anyway. So we deal with not just single strand gaps, but with anyway with double strand gaps. Yes, because Cas3 degrades both strands in the end.
Yeah. Okay, I can buy that. That's great. Thank you. Thanks for your responses and your modifications to the however many suggestions I made regarding your first draft of the PhD. I think you've done a really good job there. There's one or two minor typos and things, but we can sort those out when we discuss it later. There is one question I'd like to ask, if I can, about the response to one of my questions that I raised to the initial PhD thesis. So are we allowed to do that? Can you have a look at the suggestion 9 that I made regarding the original figure 15? Are you allowed to do that, or is that too difficult? You don't have your thesis with you?
Maybe, yes, maybe I could have a look. Yes, please, if you give me a minute.
That's fine. I promise you it's the last question from me.
Could you please remind me the picture
Well, in the original version of your thesis, it was figure 15, but I think it's become a later figure, but I couldn't find it in your new version of your thesis.
Yes, if you tell me what this figure talks about.
Yes, so the figure is, I'll read out the comment that I made. So it's comment 9 that I made. It's figure 15, whatever the figure number is now, I don't know. And
maybe it's because maybe it's about source response, right?
It is, that's correct, yeah. Can you just, I don't know if it's possible for you to talk me through that figure on screen. Perhaps you can't, but if you can't, I can ask you a direct question about that.
Oh, thank you.
Sorry to be a pain.
Okay. Yes, yes. So it's the figure which is showing the growth curves, right? It's a growth.
What is the figure number now? What is the new figure number?
It's a figure 10.
That's why I couldn't find
I believe.
Figure 10.
Yes, is it a figure which is showing growth curve in induced strains?
Ah, got it. Right, thank you. Now I can see it. Okay, so in this data, you're suggesting, you'll have to forgive me, it was a little while ago when I read this first time, but you were suggesting that this was indicative of an SOS response occurring based on these.
Yes, I changed the description indeed, because yes, indeed, it is not showing the source response itself. It's just one of the one of the hallmarks, because it's just showing that the growth is slowed down in induced strains. Yes, like it's clearly seen, but yes, indeed, we cannot judge only by this figure that we have source response. Actually, I didn't provide the figure with the DNA degradation in the PPS region in my thesis because it was demonstrated before. And in in a previous thesis of Anna Shuraeva in her article, in her paper on this model system. Maybe I should have provided it, but I think I explained it in the text.
You do, and you've cited the reference. That's good. Yeah. That's fine by me. I just wanted to clarify that because I got a bit lost around that figure. So thank you very much. You've done a great job there.
Thank you so much for the questions.
I don't have anything else I can think of right now. If something else appears, and I'll put my hand up and chip in again. So that's everything from me for now. Thank you.
Thank you.
Thank you. I have a formal question whether you are satisfied with response to your comments on the written variant and responses during the defense. It's just for the record.
Well, for the record, I can say I'm very happy with the responses I've got there from the candidate to the questions, the direct questions. Yes, that's fine. I think there may need to be a couple of typographical modifications to the amended text, but we can discuss that later.
Okay. And now, Professor Mojica, please.
Yes, hello. So first, just to congratulate the candidate for the nice work. And let me just share with you that I really enjoyed reading your thesis and also listening to your presentation. The thesis is very well written, and the experiments are well planned, and all the conclusions are completely supported by your results. So for me, the scientific quality of the work is beyond any doubt. That I would like to emphasize that even though some references in the bibliography list are still incomplete, for example, lacking final page numbers in some cases, the changes in general I recommended were made, and my questions were answered with convincing explanations. So in particular, the figures you have added are very helpful. And in this context, and probably because of the addition of these new figures and the text rearrangements you have made in accordance with the committee's suggestions, I must point out that the numbering of some of these figures, the order in which they appear in the text, so that's I believe is a minor issue. But I understand that. Still, I got a few questions to ask you, and some of them are beyond your work, the work you have done. And the first refer to the requirement of Cas3, something related to one of the questions from Dr. Ball. The requirement of Cas3 nuclease activity for priming. So I know that you didn't address this issue, but do you know if prime adaptation has been investigated in dead Cas3 mutants that is devoid of nuclease activity? In other words, has it been shown that Cas3 cleavage is really necessary for priming?
Yes, I think it was. Yes, it was also demonstrated in the experiments in our lab. And yes, yeah, it was shown to be important.
But important, but absolutely necessary.
Required. Yes, yes.
Is required.
Yes, and it is absolutely required for the assembly of the whole complex.
Yeah.
Yes.
And, yeah, but the Cas3. But what about the cleavage activity? I mean, for instance, it looks like the first nick could be necessary. I don't know for sure that. But what about the further degradation to generate the DNA fragments that encompass the region the prime acquisition complex is later bound? So that my question is more related to the fact that probably an excessive.
Yeah, I think that this nick must be important, as Cas3 may introduce this first nick, and then maybe other reg proteins may continue the degradation.
Okay, but it has not been shown that it's really needed, that nick.
Oh, okay.
I guess I was looking at the literature. I didn't find any real.
I'm not sure there was a particular experiment.
Yeah.
Yes, focusing on this.
Yeah, specifically addressing that question, because in fact, there are results that say that the nuclease activity of Cas3, I guess that makes a lot of sense, because excessive degradation of the DNA by Cas3 could be detrimental for the generation of the spacer precursors. But my doubt is more related to the first nick. Okay. Another question is that your scheme of the proposed prime adaptation model, cascade is not drawn in the translocating prime acquisition complex. That's perhaps to simplify the representation, or you think that it's not likely that Cas1, Cas2, and Cas3 remain bound to the cascade during the PhD translocation?
Yes, so just some in vitro experiments. So if I understood your question correctly, so it was shown that Cas3 may be associated for some time with cascade, but then yes, it may dissociate from cascade and continue DNA degradation in great distances. But for some time, it may remain associated.
Okay. And another question also related to your model, but it goes beyond the I-E system. You mentioned your thesis that the orientation with respect to the priming site of the protospacers sampled during priming by 1F systems is opposite to that observed in I-E systems, correct? Mm-hmm. So according to your model, as the 1E prime acquisition complex translocates, that is moving away from the priming protospacer in the 3' to 5' direction along the known target strand upstream and the target strand downstream of the PPS, the associated Cas1 recognizes the PAM complementary sequence in the other strand. Okay, that's right. That's your model. So in order to explain the opposite orientation in 1F, and always provided that the acquisition complex also moves away from the PPS in the 3' to 5' direction along one strand, at least two possibilities can be envisaged to explain this different orientation in one. So the first option is that the associated Cas1 also, as one E-system does, recognizes the PAM complementary sequence, but in the strand along which the PAC translocates, okay, instead of in the complementary strand. And the second option that comes to my mind is that the 1F Cas1 recognizes the PAM sequence rather than the PAM complementary sequence in the same strand as 1E Cas1. Which one is your favorite? Can you think of another explanation for this opposite orientation in 1F with respect to 1E?
I would more think about the first option, that the difference is just in a like complex formation in some features of the complex formed, that the different maybe the different strands are cleaved by Cas3, and that's why different strands are recognized by Cas1, Cas2.
Okay, so it's because we assume that the 1F Cas1 still recognizes the PAM complementary sequence, but it has not been shown yet. I think it has been assumed. Yes. Okay. And I just to change a bit on the context, I have also a few questions on the study of the effect of antibiotics on the CRISPR immunity in pseudomonas. So you mentioned now, and it was hard to find that in the thesis, but you mentioned that the spacer that partially matches the phage DMS genome has 5 mismatches, okay, with the target, and one of them is in the seed region. In your experiments, it's considered that the phage-resistant clones due to CRISPR immunity are those resistant to DMS vir, but sensitive to the virus that carries the anti-CRISPR, which makes a lot of sense. But you assume that the resistance to the virus, to the DMS virus, is due to spacer acquisition. And moreover, at a certain point in your conclusions, you assume that the resistance is due to prime adaptation. But however, experiments to specifically detect acquisition were not performed. That's right?
No, by the way, we did a PCR on CRISPR arrays. It was just not included here in this thesis.
Okay.
And I'm not sure it was also included in a publication. But yeah, indeed, to prove the CRISPR immunity, we did a PCR on CRISPR array, on both of CRISPR arrays. Yes, and we observed elongation. So it was like an additional proof experiment when we did it.
Okay, that's important. So you notice in all the resistant clones, in all of them, you detected amplification of the array, I guess.
Yes.
Okay, because no, I was just thinking about the possibility of mutations in that space, because just a mutation in the mismatch at the level of the seed that licenses are very strong resistance. So,
yes, sometimes we had these rare cases when like the strain was resistant to like it was sensitive to anti-CRISPR, but still yes, it was not proved by the PCR, but we don't know what happened there.
but still is related to CRISPR anyway. Okay, just reiterate my congratulations to you and on your presentation, and also the discussion, and both you and your supervisor on the work done. And I'm sure Katia would be very pleased with your achievements and the excellent scientist you are now. You have become. Thanks a lot. That's all.
Thank you so much.
Again, a formal question, Professor Mojica, are you satisfied with response to your comments to the written thesis and during the defense?
Yeah, absolutely.
Thank you. And Professor Fineran, please.
Yes, so just really like to reiterate what my colleagues have also said. Congratulations on a really interesting thesis. It's got me excited about priming again, which is always a good thing. But you've really created a really interesting body of work and very thorough experiments across a whole range of techniques as well. So really excellent effort. I thought I would ask perhaps probably some slightly broader questions, and also some that extend a little bit beyond the thesis. I thought maybe I could start also where Francisco sort of finished off around the Pseudomonas experiments. And I was just interested to hear what is your theory around how the antibiotics are affecting, the DNA substrates to then alter your space or your priming acquisition.
So did I understand your question correctly? Like, could antibiotics somehow influence the DNA substrates?
Yes, I'm thinking just even perhaps to simplify the question, do you think that this is leading to fewer phage genome copies, and therefore how could that influence spacer substrate frequency or abundance? Because to me there also seems like a question of, if you have slower phage growth, you have less phage DNA, but then you get more chances for acquisition and substrate. So they seem almost to be slightly opposite. So do you have any thoughts on that and how it could fit with your model?
Yes, so indeed, probably there is less DNA, less phage DNA is replicated when in presence of bacteriostatic antibiotics, but like in the same time, the production of mature phages happens later in time. So like the cell death or potential cell lysis happens later in time, which really provides more time for CRISPR-Cas system, which is more crucial for the successful CRISPR adaptation.
Mm-hmm. So just following on from that, can you think of an experiment that would be independent of antibiotics that might enable you to test that theory? Whether it's
yes, I didn't include it in my thesis, but another experiment was done with different carbon sources, just without antibiotics, but the cells were grown with the carbon sources which supported like slower or more or which supported or faster cell division. So when cells were growing more slowly in the presence of particular carbon sources, also the effect was similar to the presence of bacteriostatic antibiotics. So we observed the same effect, the change to the proportion of clones. So yes,
okay,
it was proved.
Yeah, no, that's a nice experiment. And I was thinking about how these results could relate to some of your work with the different genome maintenance proteins. So I imagine that if you mutate some of these genes like REC-BCD or REC-J or some of these other ones, that they might also have growth effects on the strain. Do some of these E. coli strains actually grow slower than their wild type counterparts?
Probably not this, but I would more think about the DNA in the PPS area, which maybe in the absence of REC-BCD, it is maybe it could be degraded by other nucleases more efficiently, so that like the substrates are not available for enough time. So maybe, yeah, so maybe in the absence of REC-BCD, the DNA is degraded yeah, more excessively.
Yeah.
Maybe
I mean,
less substrates are available for Cas1, Cas2 binding.
Do you know if, I mean, do any of these mutants affect E. coli's growth, or do they all grow the same sort of speed or rate as the wild type?
Yeah, we did. I think we didn't observe any effects of these mutants on, yes, on cell growth. According to our growth curves, yeah, we didn't observe any huge effects of the mutants.
Okay. Yeah, no, that's good. I was thinking if you had, whether there would be some potential influence there, but that's good to hear. So I had a couple of bigger picture questions now, and so one of them I was thinking about the fact that a lot of CRISPR-Cas systems are mobilized, horizontally. So we see them moved on plasmids, we see them transferred on, genomic islands, and phages can move them around. So they can quite quickly be acquired by different bacterial hosts. So I was just interested to hear your thoughts about what you found about the requirements of certain host factors for, assisting in substrate generation, and how that might influence the horizontal sort of histories of or ability to be acquired and picked up by different bacterial strains.
Mm.
So maybe I can word that more simply. So do you have any predictions of, for example, the I-E system? You've shown that it uses a lot of these REC-BCD and other pathways. Are there different bacteria which if they were to acquire the I-E system, they might not be able to use the same pathways of space acquisition, and therefore might not work?
It was really observed, right? It was really observed before that the REC-BCD co-occurs with CRISPR-Cas systems in genomes of proteobacteria. So indeed, yeah, it may deal also with a with supporting a horizontal transfer too.
So do you think they might have been transferred to some other strains that don't have these same pathways, but then they were unsuccessful because they didn't work, and therefore have not been retained?
Yeah, I think could be.
Yeah. So it also sort of got me thinking about some of the work around the fact that REC-BCD has also been talked about as an inhibitor of phage infection. So, this idea that the double-stranded DNA of the phage genome comes in, and that REC-BCD can start, chewing up some of that phage DNA, and therefore provide some degree of phage immunity. And people have found that, there are these REC-BCD inhibitors encoded by some bacteriophages. But I'm just thinking, most of the phages that have been studied for E. coli, they clearly can infect well in the presence of REC-BCD. So how do you think these phages are infecting well if REC-BCD is sort of, being able to access the ends of their genomes and potentially start degrading them?
I think it was proposed that this first, that the fact that REC-BCD really degrades the double-stranded DNA of the phages, which gets into the cell, it's really under question. It's very likely that it is not degrading, but more unwinding the DNA, and maybe other nucleases are degrading. And indeed, the success of the infection may be dependent on the recombination. So, like if just one phage infects the cell and there is REC-BCD, it leads to the degradation. But if there are more phages, and the phage has a template for recombination, and it may survive after the targeting by cellular nucleases. So the success of the infection may depend on the amount of the phages which are infecting the cell be simultaneously.
Yeah, no, that's an interesting idea. So I was also interested in your model where you're targeting using the self-targeting spacer to generate new immune memories. And I was wondering, kind of following along the same theme we've just been talking about, thinking about phages, whether you think you would get similar results if you were to look at spacer acquisition from a incoming phage compared to this targeting of the bacterial genome, and maybe just to sort of add an additional factor in there is, something you mentioned as well, is that I guess phages often carry many of their own recombination proteins and things as well to help, resolve some of their genome intermediates during replication and things like this. So I guess sort of two parts to the question, do you think you would get similar patterns from phage DNA? And secondly, do you think any of these phage recombination proteins could also influence spacer acquisition from a phage?
Yes, it's a very interesting question. And no, for sure, I think that the effects would be different just because of the size of the DNA which is targeted. I think we will not even observe the same pattern, maybe like the same extensive pattern of spacer acquisition, like in our model, because the patterns will cover each other, like they will. Sorry, I cannot find the proper words. So yes, there will be differences, of course, and also like because of the different velocities of replication, and I think that the roles of REC-BCD and REC-J, which we found, they should remain in with any type of DNA target, but like the amount of the observed effect, like the scale of the effects should be different. I think.
Yeah, and then any do you have any thoughts on whether phage recombination proteins could contribute or interfere or somehow be hijacked to promote?
Yes, sure,
things?
it could be, of course. Yes, it's it's a very interesting topic maybe for future studies. Yes.
So I think I have no further questions. So just thank you again for a really interesting thesis, and congratulations on the work.
Thank you.
Okay, thank you, Professor Feineron. And again, the traditional question, whether you are satisfied with responses to your comments and on the written text and during the defense.
Yes.
Okay, thank you. And now, Professor Khrameeva, please.
Congratulations for the excellent work, and thank you for the opportunity to read it. Actually, I'm not an expert in the field of your particular thesis, so my comments and questions are rather technical and are about the data analysis and about the data sequencing part and general statistics. maybe some of them are less important than others, and I've seen the updated version of the thesis that you submitted, and I'm happy actually with all the corrections that you did. So maybe we could discuss just a couple of my questions that are maybe, in my opinion, are more important than the others. So one of the concerns was about the averaging the replicates of the experiments, and as I wrote in my jury member report, it's totally fine to take an average of the experiments and continue the analysis based on the averaged value. But before doing that, it's important to demonstrate that the replicates, that the experimental replicates are actually similar to each other. So yeah, maybe you could comment on that.
Yes. So in this, as I replied, the effect, yes, the effect was many times proved in our lab and later proved by the results using different methods like fractate data and HIPS data analysis completely proved the results on the first work. I indeed agree that the demonstration of the that the replicates coincide is quite important. We didn't, as the graphs are really almost overlap for replicates. We didn't think about the importance of providing the information about the compatibility. Yes, later when in the following work, in the next work, we did the analysis of more mutants, of a big amount of mutants, including the same mutants, and the results were approved. Yeah, so I included examples of analysis for other mutants. But yes, I agree. In later works, we should also, yes. So maybe also two replicas, as it was just a pilot experiments. Just two replicas were enough then, but it's better to continue with more replicas in the future for sure, like it was done in following work.
Okay, this is fine. And another my question was about the normalization of the data that you used in the methods. I couldn't find the details of the normalization procedure. In particular, it wasn't clear whether you somehow normalized for the total library size. I mean, when you sequence samples, just because the sequencer produces different number of reads, you can get different coverage for the samples. So did you normalize?
Just usually we didn't compare quantitatively the data. When we analyzed the patterns of acquired spaces, we were not even interested in the efficiency, like in the amount of spacers, because we were comparing only patterns. We analyzed the efficiency using gel bands or yes. And yeah, I think the normalization was done when we compared the coverage of by short fragments. And yeah, there I believe it was done. It was not my responsibility in this work. But yes, in that case, it was important.
Okay, so this part of data analysis was done by someone else.
By Anna Shirayeva, yes.
Yeah, I see. And maybe the last question was about the figure 27. It concerned the choice of the statistical test to assess the statistical significance. You chose Welch's t-test, and yeah, but you actually formally did not demonstrate the normality of the data, the normal distribution of the data, which is one of the assumptions of this particular test, and formally it's required. So I just recommended to use Wilcoxon test instead of Welch's test. But yeah, it's, I think, a minor thing, and I have little doubt that the choice of the Wilcoxon test would influence on the conclusions of the thesis that you present in general.
Yes, so in the end we checked the normality and with additional test, and it was proved.
Yeah, then I think it's fine. And other my comments, they are regarding some suggestions about improving formulations, some things in the text. So they are about the writing part, and that they were corrected. Yeah, so I don't have other comments on my side.
Thank you.
Okay, and again, the formal question, whether you're satisfied with the responses.
Yeah, I'm quite satisfied with the responses. Thank you.
Okay, cool. Thank you. Sorry, Professor Kotelevtsev.
Elena, thank you very much. I thoroughly enjoyed now your presentation, much more than when reading, because you introduced slide number 16. Thank you very much, explaining where these spacers are coming from. Because first, as we discussed after the reading, it wasn't clear for me essentially where these spacers are coming from, but now you introduce this slide, and I understand that this is a kind of a self-immunity. You induce the Cas system and recognizes the sequences very bad. So this is much more clear for me, and I'm completely, and this was only one my remark, and I asking the formal question of Mikhail Sergeyevich, I'm completely satisfied. Now I have a couple of questions arising from what you were presenting today. And the first question is, you mentioned at around slide 36 in conclusions that you were not able to detect any effect of genome maintenance proteins on the processing of 3' end of the spacer. Is it right?
Yes, yes, indeed.
Okay, and if it is right, the question is, what are the potential candidates which you envisage, which might be analyzed? Because you used essentially, as we call it in genetics, candidate gene approach. You appointed the candidates like RABCD, REG-J, EXO7, and you explored the effect of these candidate genes which are involved in genome maintenance on the acquisitions of the spacers. What your approach will be if, say, you will continue this study and would like to find the genes which affect the processing of 3' end of the spacer?
So one of the potential proteins yes, so yes, in this work we were mainly focused on 5' end processing, and in vitro, in other CRISPR-Cas systems, there are proteins which were demonstrated to suggested to take part in 3' end processing. But in our experiments, in our preliminary experiments, we couldn't obtain the similar results in our system. So yeah, we're still working on it.
But just off the head, could you name some candidates which might be involved in this process? Maybe other genome maintenance genes which were not explored here, not REG-BCD, not REG-J, not EXO7. Are there any other genome maintenance proteins in
It's
E.
like
coli?
quite a lot of many others, like DNAQ, and
So the
there
list
are quite a
is
lot
huge.
of them. Yes,
Okay,
the
then,
list is
so
huge.
you will not be short of the list nominating another kind.
Not
Good.
so. Yes,
I
as
understand that.
I told,
The last-
like
yes.
the short list which was suggested by other works was not proved still in
Yeah.
our experiments.
Okay. And another question, are there any experimental evidence that actually the proteins in question, which you investigated, like REG-G, REG-BCD, and EXO7, physically interact with the protein complexes of cascade gene or acquisition? Maybe there are some experiments in to hybrid system experiments. Do you know anything about that, or have you approached this question?
Indeed, Cas1 was demonstrated to physically interact with REG-BCD and SBCB. We didn't find any effect of SBCB mutation in our work, surprisingly, but it was demonstrated to interact with Cas1. And the demonstration that REG-BCD interacts with Cas1 adaptation enzyme, was really supported in our experiments.
Yeah.
Yeah.
So it's a published work from.
Yes. It's quite old work, by the way.
Yeah. Okay. Thank you very much.
Thank you so much.
I'm satisfied completely.
Okay. Thank you. And as for myself, I've had mainly editorial and technical comments. Also, maybe some of the comments intersect with what has been discussed already. So I think to save time, we'll skip on that. The changes and the responses were completely satisfactory. So you're done with me, basically. Okay, thank you. Are there any questions from the audience here in the room or in Zoom?
Meanwhile, I can see no questions in Zoom.
Okay, so I see none here either. So I think we may move on. And Professor Severinov, please, the supervisor.
Thank you. Am I supposed just to talk on Elena as a student or on the work, scientific work?
Technically speaking, I think it's about her as a student, but we won't. I mean, if you want to comment on the work as well, that would be fine.
No, so yeah, I was time flies. So I was surprised when Mikhail Gelfand mentioned 2014. So I did a small check on my email, and indeed, Elena approached me in the summer of 2014. She was a third-year student of a 6-year program, specialist program in Moscow State University on bioinformatics and bioengineering, where she said that she would like to do CRISPR. So in 2014, it was still almost pre-scientific to do that. So that shows her good judgment. And also, my experience is it's always the students who come to you as opposed to the ones you run after that would be the best ones. So she proven that to be the case. So she started her work with Katya Savitskaya, the late Katya Savitskaya, working on something completely different, mostly on phage responses to CRISPR. And she then decided to stay in the lab and was in the master program of Skoltech, where yet again she was doing something different from what her PhD thesis ended up being, but in a way related. And I'm really sorry that that has not been published at least yet. So she was working in an academic mobility with Udi Kimran on, believe it or not, the effects of GP 5.9, which is a T7 DNA mimic protein as we know it, on the inhibition of CRISPR adaptation, naive CRISPR adaptation, which of course back then we didn't know that GP 5.9 also inhibits REG-BCD. In a way, it would have been a nice addition to her thesis, which already has 4 different chapters, but that could have been something which would combine some of these topics in a very nice way. And I hope that paper would still be published. And so I think she's great, and she is one of the still few, unfortunately, people who can combine experimental work, like much of the strains she was talking about, she made herself, as well as advanced bioinformatic analysis, whether with or without Wilcox test. So I think she certainly, if I'm supposed to say what she deserves, then I think that she'll be an A, and clearly a degree that she's seeking. She really was supposed to defend a year ago, were it not for the war. So there was this hiatus. but I'm glad that this is now being brought to a closure, and I wish her well in her future endeavors. I think she's a great scientist.
Thank you so much.
Okay, thank you. Any other matters to discuss? I see none, so probably we have to ask. Yeah, we have to ask the student,
To leave.
the audience to leave for a moment. We'll discuss.
So yes, dear colleagues, I will now open a separate breakout room only for the jury members who attend online. All the rest of the audience is most welcome to stay connected. We will be back after the jury discussion for the announcement of the result.
Elena, so the members of the jury discussed your thesis and your presentation, and everybody valued highly the quality and significance of your work. However, several members of the jury had concerns about like misprints, missing references, and minor things like that. So after the vote, the decision of the jury was accept with minor changes, pass with minor changes. That means that you have to correct like the remaining typos and the remaining small things. You don't have to, well, you'll submit your written thesis, but you don't have to go through the entire procedure. So after you submit your thesis with the required changes, you'll get the comments of the jury members. You will automatically become, after approval, technical approval, you will become philosophy doctor of Skoltech. That's it.
Thank you, dear jury members, and thanks to Skoltech. I was happy to be a PhD student in Skoltech and conduct the work I presented. Thank you so much.